Thank you. As I was just mentioned, I'm at Cornell University in Ithaca, New York, in the Department of Molecular Biology and Genetics. But I'm also director of the Center for Innovating Neuroimmune Disease. We currently have a major collaboration with the Hospital for Special Surgery in Manhattan, New York. We're actually 4 hours northwest of New York. We have 4 faculty groups at Cornell and Ithaca who are currently carrying out research on ME. And we have 2 faculty groups at the Hospital for Special Surgery. And this photo shows the people who are involved in our studies. Not everyone was there, so we had to Photoshop a few people in, including me. So my disclosures are funding sources for our center here, and I'm also on the scientific advisory boards for 4 ME foundations. So I'm going to start by discussing T cells, but I will mention some other immune cells as well. But T cells normally alter their metabolism in response to foreign antigens, and there are 2 main groups called CD4 T cells and CD8 T cells. And both of these are very important for proper response to an infection. But I'll be concentrating a lot later in my talk on the CD8 T cells, which secrete cytokines but also cause death in cells that are infected with something, or cancer cells. But T cells can become non-functional, so-called exhausted, by chronic exposure to a viral antigen or a cancer antigen. This diagram here shows what happens. So if you have a cell that's infected and it may present an antigen from the virus that the T cell can then detect, and when it detects it, it will do a cytotoxic attack on that cell and get rid of it. But if there's exposure to chronic infection, then the cell's gene expression changes, and inhibitory receptors expressed on the T cells that blocks this cytotoxic effect. And while this can result in the persistence of a chronic infection, it may also be protective to prevent so many cells from being killed in one's body that you are killed as well. So, so this is a very important phenomenon that we need to understand more about. We have published 2 papers from my lab in which we looked at T cell metabolism. And in these 2 papers, we suggest that the T cell metabolism abnormalities we detect hints an exhausted state. So what are these hints of an exhausted state? Well, T cell metabolism normally changes following stimulation of the T cell after it's detected this foreign antigen. And the T cells start out being quiescent, but when they are proliferating because they've been signaled to proliferate because of detection of a foreign antigen, the glycolysis is upregulated, oxidative phosphorylation is upregulated. In contrast, the quiescent cell is using more fatty acid oxidation for its metabolism. But when the immune system is being chronically stimulated, these metabolic responses don't occur properly. So I'll summarize some work here. This figure actually came from a talk given by my former student, Jessica Maya, who is here in the audience. She gave a talk at that roadmap, one of the roadmap webinars that Vicki Whittemore mentioned earlier in this session, that you can actually look online and watch this webinar and many others in the metabolism webinar of the roadmap. But what was summarized here is in the CD8 T cells, shown in blue, glycolysis we found was reduced in the cases versus the controls, as well as the mitochondrial membrane potential, and fatty acid oxidation was increased. And these are all hallmarks of exhausted T cells. In addition, we saw that higher plasma IL-10, that's a cytokine, correlates with lower glycolytic rates in ME. So the worse your glycolysis was in your CD8 T cells, the higher the plasma IL-10. And that's a cytokine that limits response to pathogens. It's a immunoprotective cytokine, immunosuppressant. So there's another report that hints at an exhausted state of the immune system. As I mentioned, those two reports couldn't absolutely conclude that there's T-cell exhaustion, nor can this one. But this one also is very interesting with regard to the possibility of T-cell exhaustion. And one of the authors is also, two of the authors are actually here in the meeting, Mady Hornig and Nancy Klimas. So in this paper, they studied production of cytokines. So when foreign antigens detected, CD8 T cells are sent signals by other cells to produce certain cytokines in order to aid in defense against whatever is invading. And two of these cytokines are interferon gamma and TNF alpha. But again, this response may not occur if the CD8 T cells are dysfunctional. So this is a figure from this paper that you can consult in the journal shown. And fewer T cells from ME and also long COVID subjects, which are studied, responded to the stimulation with production of cytokines, interferon gamma and TNF alpha. So this is the stimulated long COVID cells from long COVID patients and from ME patients in comparison to controls. And you can see here that a lot more interferon gamma was produced by the healthy controls. And the same is true for this TNF alpha, that more TNF alpha was produced by the cells than the long COVID cases or the ME cases. So this again is some evidence that there could be T-cell exhaustion occurring. Now, earlier in this session, Brian Wallet also presented results from the NIH intramural study. He showed two assays that suggest that CD8 T cells are dysfunctional, and that again is additional hints that T-cell exhaustion may be occurring. But again, it's not adequate to prove that T-cell exhaustion is occurring according to an immunologist. Immunologists have told us that you need to have really quite a lot of evidence in a number of different methods in order to conclude that there's actual T-cell exhaustion. There's other phenomenon that can occur as well. So analyzing, we went to analyze specific types of immune cells, and because we think this has been critical for understanding immune dysfunction in ME, rather than just studying all, total PBMCs or total cellular contents, that you need to really look at individual cells in order to dissect what's happening in ME and in other diseases as well. So this is a report that recently appeared that was spearheaded by Andrew Grimson's lab. And his lab did single-cell profiling of immune cells in ME versus sedentary controls. So all of our controls were selected to make sure that they were no more physically active than the ME cases that we studied. Obviously, we could not study severe, completely bedridden cases, but most of our cases were moderately ill. So 30 ME subjects and 30 sedentary control subjects, cells were put through a device that separated each cell and then could provide data that, on the RNAs present in each one of those separated cells. So once you get the data, you can use the single-cell sequencing data to identify the type of each cell whose RNAs were sequenced, because previous studies in which these cells were isolated and then their RNAs profiled, we have a good fingerprint for each type of cell, and so you can use techniques to group the cells of different types, such as a monocyte or a natural killer cell, T cells, B cells, and all the other cells as well. So this is a very simple diagram here. It's actually more complex when you show it in the actual data. Here's an example. Here is the actual data and displayed like this. Each little dot that you see here corresponds to a single cell. So here, these are all B cells. These are monocytes up here. These are naive CD4 T cells, etc. So the different type of T cells could be identified and then mapped on this using an algorithm on a graph such as this. So the type of cell with the most gene expression differences was then detected to be classical monocytes. So all those different groups that I showed in that previous slide are shown here as these numbers. And the ones with the most, this is the number of dysregulated, differentially expressed genes between cases and controls. And this group 2 corresponds to classical monocytes. The red here shows the dysregulation, the difference before these patients performed a maximal CPET, and this is 24 hours later. So you see that the maximal CPET actually didn't change the number of dysregulated genes very much in this particular case. So these monocytes were already dysregulated before the exercise was done. So to sort of summarize what I'm going to be telling you, I have this nice diagram done by Jessica Maya showing what classical monocytes do in ME/CFS subjects. They express genes indicating they're being signaled to develop into macrophages. And so what are macrophages? Well, this is a type of cell that migrates to tissue and surrounds and kills pathogens or infected cells. So this makes you suspect that something is, what is telling them to do this? Why are they doing this? So in order to, when you do single cell RNA sequencing, there are 30,000 genes in the cell. You don't get the RNAs from all 30,000 genes. Technology is not sufficient to do that. So in order to get more RNA information, Andrew Grimson's lab purified classical monocytes from 4 cases and 4 controls, and then did total RNA-seq, bulk RNA-seq of all of those cells, and then graphed here the more difference between the patients and controls, the higher the number here. And then on this y-axis, we're showing the different proteins that were detected. So this one here, this CSF2, is also known as GM-CSF. It's a cytokine that is normally known to stimulate cells to produce granulocytes and monocytes. And these 2 chemokines are known to attract immune cells as they go through the endothelium and migrate into peripheral tissues. So this was consistent with the idea that there's some signaling going on to make the monocytes go into tissues and become macrophages. What's interesting is this increase in GM-CSF has been seen before. Here's a study from Montoya's group showing it's actually higher the more severe the patient is. And a study that we did with the Columbia group, Mady Hornig and Ian Lipkin and their colleagues, we also saw that the sicker the people were on the SF-36, the lower the score, the higher the GM-CSF they had. And I always like to see correlations because I think that tells you something about the illness, to see a correlation. And something that a talented student did using machine learning, developed an algorithm to classify the individual classical monocytes as to how weird they were in comparison to normal control monocytes. And the controls, then the number, these were so-called diseased monocytes. Most of the controls had very few so-called diseased monocytes, while most of the cases had quite a number of these, quote, diseased monocytes, although you can see that there's some variation in the range of the number of monocytes that looked unusual and abnormal in the cases. But again, what was particularly striking is that the higher your score on the MFI fatigue inventory, the higher the number of diseased monocytes you had. So this really gives some confidence that having these abnormal monocytes has something to do with the fatigue of the illness. So this has all been published in that paper I indicated, but we've now gone on to do some additional analyses that gets back to this question of T-cell exhaustion. And a lot of the work I'm going to be talking about, sequence analysis, was done by David Yu in the Grimson lab. So just like this is a graph again showing whether what the abnormalities are in those different groups that I showed earlier. And so there are some differences where the cases are upregulated genes or the cases are downregulated. But what we see a lot of difference in, again, is the CD8 T cells. So those CD8 T cells have a lot of difference among the T cells. So in order to study this more, again, you want to get more sequence. So the CD8 T cells were isolated, and we wanted to analyze two types more thoroughly. There's so-called naive cells, which haven't been exposed to an antigen, aren't yet responding to an antigen. And then after the naive cells are exposed to an antigen, they become effector cells and then memory cells. So they become cells that actually can respond to antigens. So the CD8 T cells were isolated again for deep RNA sequencing from total PBMCs. In this case, we used 7 cases and 7 controls from our cohort. Used flow cytometry to separate the cells into naive cells and antigen-experienced cells, ones that had been exposed. And then looked to see, is there a difference in the different number of differentially expressed genes between cases and controls? And indeed, the experienced cells had more differences in their expression than these naive cells. So then the idea is to look closely at the genes that are differentially expressed. And I'm just going to show you the ones that we're particularly interested in because they are relevant to T cell exhaustion. So these genes that are associated with an exhausted state were upregulated in the ME antigen-experienced cells. So here's these 2 genes called SLAMF6 and SLAMF7 are genes that are known to inhibit the action of T cells. They're present when you have an exhausted state. And this EOMS is a transcription factor that's also characteristically upregulated in exhausted cells. Another interesting aspect of the gene expression was actually metabolism. So it was this is the hexokinase gene here. So this hexokinase is one of the genes needed for glycolysis. It's lower in ME, and as I showed you earlier, we found that in the ME cases, CD8 T cells had lower glycolysis, and that is a characteristic of T cell exhaustion. Another interesting thing is that there was upregulation in this glucose transporter, and that is a characteristic of exhausted T cells. So this encodes GLUT1, this gene encodes GLUT1, which is a transporter for glucose. So another way of looking at this is not just looking at the gene expression, but also looking at proteins. And this work was done by Jessica Maya in my lab, in which she isolated specifically CD8 T cells using a magnetic bead, using magnetic beads, and then put these through flow cytometry to look specifically at some of these factors at the protein level. So you heard Brian Wallet mention this PD-1, which is upregulated in some cells, but also, and it's a sort of very, very well-known marker for exhaustion that is often found in cancer, and there's work has been done to modulate this PD-1 in cancer. SLAMF6 is also an inhibitory receptor, and then there's this TOX transcription factor that is also known to be upregulated in exhausted cells, and in fact responsible for some of the exhaustion gene expression. As shown in, again, this nice diagram produced by Jessica Maya, which shows what the action of this transcription factor TOX. So it can alter the expression of genes, resulting in the sequence in the expression of RNA that then results in the production of PD-1 and other immune suppression markers, and in absence of this activation, you have a decreased immune response of as well. It can turn off some genes too. So these exhaustion markers in the cells, these at the protein level, are also elevated in cases versus controls. And this is looking at total T cells rather than separated ones. But again, the PD-1 is elevated, the SLAM-F6 is elevated in the cases. And then if you look specifically at T cells that are carrying PD-1, they have elevated SLAM-F6 in the cases. And then looking at the exhaustion-associated transcription factor tox that I mentioned, again, looking at the cells that are antigen experienced that have developed, one sees that the tox is expressed, tox transcription factor is expressed at a higher level, and also the T cells that are expressing PD-1, the tox is at a higher level. So in addition to seeing this higher expression of tox at the protein level by flow cytometry, the Grimson lab has also used an epigenetic method called ATAC-seq to identify regions of open chromatin. Now you don't need to know how this method works, but what it can do is measure how open the chromatin is. If it's open like this, then transcription is on. So you have an increased expression of genes when you have open chromatin versus chromatin that's not opened up. And what his group has found is that 4 different regions near the tox transcription gene were found to be more open in the ME cases than controls. So this is that data. These are 4 different regions, and in the cases, there's a higher level of accessibility of that region. They're more open in these 4 different locations. So this again indicates the change in the gene expression consistent with exhaustion. So to summarize here, I've shown, this is a diagram from a review article, but I just want to review what we now know about the evidence for exhausted T cells. So as I mentioned, there's this decrease in interferon gamma and TNF alpha, an increase in GLUT1 expression, a decrease in glycolysis, an increase in fatty acid oxidation, and lower mitochondrial membrane potential, increase in tox, increase in SLAM-F6 and PD-1, all characteristic of exhaustion of T cells. And so there are antibodies against inhibitory receptors that can mitigate this T cell exhaustion. And this is shown here, that there are antibodies that can bind to these inhibitory receptors and then restore the function of those T cells. And that's been widely used in cancer treatment. So there are FDA-approved drugs to do this. So just to end, I would say, what can be stimulating the immune system in the pre-2020 ME/CFS, which is the subject of all our work? There could be an undetected chronic infection by an inciting virus that we don't know about. There could be known chronic infections by herpes viruses that are reactivated that are causing this T cell exhaustion. There could be abnormal expression of endogenous retroviruses, something we heard about yesterday. Or maybe there's actually a normal human protein like a cancer antigen that following an infection has become recognized by the immune system and is causing a problem. I'll just end here with my acknowledgments. Thank you.